And hopefully you should all see my first slide. Okay, we're going backwards. Okay, so this is part 2 of our mass spectrometry training series, and today we're going to look at protein analysis using tandem mass spectrometry. So last week we looked at how mass spectrometers work and what mass spectrometry is. Today we're going to look in detail at how they can be used for analysis of proteins. So this, you may remember from last week, was what I described as a black box approach. We have sample preparation, samples going into the mass spectrometer. Here we have the mass spectrometer, which we said we could describe that as a black box, and then we have data analysis. Following last week's talk, hopefully you'll get some idea of what's going on inside that black box. So protein analysis is probably one of the major uses of mass spectrometry. It's mostly done by tandem mass spectrometry or fragmentation mass spectrometry. And I'm going to focus first of all on the sample preparation. Sample preparation quite often involves a large amount of work, and it is absolutely critical to the success of almost any mass spectrometry experiment. So we'll focus first of all on sample preparation. So the first thing to understand about mass spectrometry is a phenomenon called ion suppression. It's one of these things that you probably won't hear very much about, but what it means is that the ionization process, particularly in electrospray, is competitive. So if you have different species in your electrospray plume, some of those species will compete with other species for protons, and some will be successful and others will be less successful. And this is a phenomenon of ion suppression. So in mass spectrometry, very often what you see is not necessarily what's there. What you see is what ionizes preferentially. And the corollary to this is that mass spectrometry works best when whatever it is you're analyzing is pure. Now, in a biological context, that is almost never the case. Also concerned with the biological context is that mass spectrometry and electrospray in particular is incompatible with most biological buffers. So the contents of these buffers, these are salts, detergents, non-volatile compounds such as glycerol, are fundamentally not compatible with mass spectrometry. So what can we do about this? Well, the solution to this has been around for a long time, and the solution is either reverse phase Hplc purification or SDS page purification, or very often both of these, which function to desalt the sample, to remove small molecules, and crucially, to separate the components of a mixture so that they can be delivered into the mass spectrometer one component at a time. And mass spectrometers, electrospray mass spectrometers, are always connected to an Hplc. And the reason for this is for sample cleanup, for chromatographic separation, and also for automation, which is extremely important. And this connection between an LC system and a mass spec, this is known as hyphenation. There are different types of hyphenated techniques. The most common one is LC-MS or LC-MS-MS. And what you need to understand is that the better the chromatography, the better the results you will get from your mass spec experiment. So just to say something about reverse phase Hplc, if you're not familiar with it, reverse phase Hplc is a very, very common separation technique. In this case, in the example I've shown you, peptides are eluted from a column using an acetonitrile gradient. There are huge advantages to using Hplc reverse phase. One is it gives you very, very good desalting, and that's essential for successful electrospray. HPLC has the highest peak resolving power of any liquid chromatography method. So that's also important because we want to separate very complex mixtures. There are two mobile phases because we generate a gradient, and both of those mobile phases are volatile. And that's important because they're going to be fed into the mass spectrometer. They need to be mass spec compatible. That's not the case for all LC buffers. For example, if you're doing ion exchange, the buffers are not mass spec compatible. It's also important to understand that mass spectrometry as a technique, the acquisition of mass spectra is extremely fast. This can happen in microseconds. It's not generally viewed as a fast technique because we have to feed the sample into the mass spectrometer by HPLC, and in comparison, that is much, much slower. So a typical HPLC separation might take several minutes, or it could take 1 or 2 hours. However, over the last 10 or 20 years, there have been enormous improvements in HPLC. There's been the development of nano HPLC, of ultra high pressure liquid chromatography, special monolith columns, and particularly UHPLC and monoliths have enabled the HPLC to be done much faster. And this is just an example of an HPLC separation for peptides from the Herceptin antibody, and just to give you some idea of how good that separation is. So we want to use mass spectrometry to identify proteins. We cannot generally use the protein mass, and the reason we can't do that is because the mass of a protein in general is not known. And the solution to that is we use Tandem MSMS, fragmentation mass spectrometry. We fragment the protein in order to generate sequence data. And it's the sequence data that we can use to identify the protein from a database. I want to give you an example now of a protein that we could do some mass spectrometry on. This is SLC13A5. You can see from the UniProt entry that it is a membrane protein. Okay, that has important ramifications. It's a membrane protein. So here's another page from the UniProt entry, and it's given us a mass. Okay, this mass here of 63,062 daltons. However, that mass is derived directly from the genomic sequence for SLC13. And that mass is nearly always wrong. I almost wonder why they bother putting it there, because it is virtually no use. And the reason for that is because the genome is not the same as the proteome. And if we look further into the UniProt entry, we can see that SLC13 is glycosylated. And that's not unusual. The post-translational processing for proteins is usually highly variable, and it's also usually unknown. And unless you know that, you can't predict the mass. So you can run proteins on STS page. STS page, you will derive the mobility, and from the mobility you can calculate the apparent molecular weight. Now remember, this is a mass spectrometry seminar. We do not measure the mass from STS page. You measure the apparent molecular weight, and this is based on its gel mobility, which is not necessarily perfectly perfectly matched to its mass. So the apparent molecular weight is approximate from a gel. It's typically plus or minus 5%. However, proteins do not always behave normally on a gel. So on this gel, I boxed in, this is real data. This is SLC13 gel. I boxed in the SLC13 band, and you can see that it runs faster than the predicted molecular weight. So observed mobility is at 50 kilodaltons. This is not unusual for membrane proteins. One feature of membrane proteins is they always run on a gel faster than expected. And you must please remember that proteins can never be identified on the basis of STS page alone. Lots of people do that. It's a mistake. It's a gamble. You cannot be sure of the identity of your protein by STS page. So another problem we have is that of sensitivity. So mass spec sensitivity is best for small molecules, for small, at the bottom of the MZ range. So the sensitivity is best for peptides and small molecules. It's less good for intact proteins. So the solution to that is, and also proteins are very difficult to fragment. So the solution to that is we use a specific endoprotease. That protease will generate predictable peptide fragments from the sequence. So this is the sequence for SLC13. And I've annotated this with the triptych cleavage sites. So trypsin is the most commonly used endoprotease. I've marked here the triptych sites, which are lysine and arginine. So as I said, trypsin cleaves at lysine and arginine. If there's a proline before those residues, it will not cut. Trypsin is an excellent reagent. The specificity is extremely high. You can buy special proteomics grade trypsin, which has been dimethylated on its lysines. This prevents its autodigestion. It's very stable, it's very cheap, and most importantly of all, trypsin cleaves typically every 10 to 15 residues. And this size is ideal for peptide MSMS. There's an optimal protein to enzyme digest ratio of this, 20:1. The incubation has to be above pH 7, and it's typically done at 37 degrees overnight. There are ways of speeding it up slightly, but nevertheless, the digestion is usually the slowest step in your analysis. So we can do an in silico digestion of SLC13 for trypsin, and this is what we've done. There are no miscleavages, so the assumption is that the digestion has gone to completion. So we have a range of peptides, theoretical peptides of SLC13, and I've marked them here. The ones that I've marked in yellow, like this one, FK, you can detect that. The problem is it's too short for that sequence to be statistically valid. To be statistically significant. So you can't use that. It's not useful in a database search. The ones I marked in orange, these peptides are long enough to give you statistically significant data, but they are too long to readily fragment. So generally you will not get any signal for these peptides. So the remaining peptides, these are the ones that we would expect to see in our MSMS experiment. There is another problem, and that's the problem of disulfide bonds. If we imagine we have a peptide sequence here with, it has some lysine and some arginine residues. If we digested that, it would generate these three peptides and these two single arginine residues. Sorry, two peptides and these two single arginine residues. However, we have some cysteines in this sequence, and if those cysteines are disulfide bonded, that digestion will look different. It will now look like this. So instead of having two peptides, we actually have a single peptide joined by two disulfide bridges. The problem is we don't know that in advance. So we will not have a predicted mass. So we cannot match that in a database. Also, this peptide is now considerably bigger and it may not readily fragment. So we need to do something about cysteines, and we need to break those bonds, and we do that with dithiothriotol. In order to prevent those bonds from reoxidizing, we will alkylate them with iodoacetamide, and that will form a carbidomethyl cysteine modification, and that's a global modification. So every cysteine in our sample will be modified to carbidomethyl cysteine. There is another problem. The cleavage sites, they can either be too close together or too far apart. And the solution to that problem is we don't just use trypsin. Trypsin is nearly always your first choice. I would say that probably 98% of all the analyses that are done are done using trypsin, but there are alternative enzymes. In our lab, we would typically use chymotrypsin, pepsin, or elastase, or we would try and aim for a partial digestion. Another problem is those cleavage sites may be inaccessible. And just to go back to our example of SLC13, it's a membrane protein. As I said, I have marked on here the membrane spanning regions of SLC. In those regions, there are no charged residues. Trypsin requires lysine or arginine, which are positively charged. So there are no cleavage sites for trypsin. So this is some real data. This is some MSMS data for SLC13. We have quite low coverage, only 8%, but that's plenty of coverage to allow us to identify the protein. But you can see that it's quite restricted. If I superimpose on that sequence the membrane spanning regions, you can see that in all examples apart from here, we only get sequence data, we only get fragmentation data where there is no membrane spanning region. Okay, so that's a common problem with membrane proteins. Basically, the enzyme does not get a chance to work. So the solution to this is denaturation. We can denature with various ways. We can denature using heat. We can denature using acetonitrile. We can use a high molar concentration of urea. We can also use a thermolabile detergent. The reason it's thermolabile is whatever denaturation step you use, it has to be compatible with your protease, and it also has to be compatible with mass spectrometry. So if you're going to use urea as a denaturant, you must dialyze it away before you complete your analysis. So despite all of that, peptides will still not be found if we don't allow for missed cleavages, okay? So you cannot assume that your digestion is going to be complete. So it's normal to allow for 2, 3, 4 or more missed cleavages in your digest. Your peptides may be modified. If they're modified, we are not going to know the mass. You're not going to identify your peptide if the peptide yield is too low. You may struggle if your sample complexity is too high. And also, you must remember that if the peptide sequence that you have data for is not present in the database being searched, you will get no result, okay? And it's important to remember that. It's very easy to assume if you searched a large database and there is no result, you tend to assume that there's nothing there. But it may well be that the sequence you're looking for is not actually represented in the database. So remember this, because this is true for all types of mass spectrometry. Absence of evidence is not the same, it's not equivalent to evidence of absence. So we can prove that something is present in mass spectrometry, but we cannot prove that it's not present. So Tandem MSMS is an extremely useful technique. It's very, very sensitive and it handles complex samples extremely well. However, you should remember that there are problems with Tandem MSMS. One of them is the MSMS protein sequence coverage is always going to be incomplete. Not everybody you talk to will tell you that, but believe me, it's true. You cannot guarantee 100% coverage of your protein. Because of that, your MSMS data, it does not give you the full protein sequence. It allows you to identify protein homology. A certain proportion of protein sequence will be matched, but it will not give you the identity of the protein. You can only match by homology. Also related to that, Mascot, which is the most commonly used search engine, is extremely poor at differentiating different taxonomic species. It's also extremely poor at differentiating different protein constructs. If you do use it to analyze different taxonomic species or protein constructs, you will find that protein homologs hits with the same mouse score, they will be listed by the program at random. So everything with the same score, it just listed at random. So if something is at the top of the list, it doesn't mean anything. Another thing about MSMS is it cannot differentiate between intact and between partially degraded proteins. Nor can it differentiate between isoforms resulting from post-translation modification. Just to kind of illustrate that, these isoforms of this protein are different. However, when you digest them with trypsin, they will look exactly the same. Okay, this isoform here is not the same as this isoform here. But by MSMS, you will not be able to tell. So let's have a look, so that's sample preparation. Let's have a look at what's actually going on inside the mass spec. Okay, so we need to load our sample. This is done, this is the auto sampler viewer. We can load our samples either in vials or in microtiter plates. In this example, we have 96 well microtiter plates. So we have the ability to load a very large number of samples and do automated analysis. And we need to program the mass spec. So we need to basically tell it what to do. And there are various things that the LC-MS system needs to know. First thing it needs to know is where is the sample. Okay, so we have a column instruction here telling it this is the location in the auto sampler. This is green sector, plate C, row C, well number 2. So this tells you where the sample is. So it needs to know where the sample is. We need to call the sample something. So what is it? What is our named sample? It then needs to know it's going to generate data. The data files are quite large, and we need to tell the instrument where that data is going to be stored. And this is not really a trivial thing, because you will find that in mass spectrometry you can generate hundreds, thousands, or in our case, hundreds of thousands of files. And where you store it matters, and how you store it matters, so that you can retrieve data, historical data. If you can't retrieve it, there's no point saving it. We need to know which Hplc method is going to run. Remember, we have an Hplc coupled to our mass spec. It needs to know what gradient and what flow rate it's going to run. We need to know which mass spec acquisition method we're going to use. We can set up the mass spec to do different types of experiment. It needs to know which one it's going to use. When the data emerges from the mass spec, it needs to be processed. So we need to do spectral processing. I'll explain how that works in a minute. And then finally, we have a database that we want to search. So we have to specify which parameters we're going to use in our search, and also which databases are going to be searched. So this is our Hplc method. Your sample needs to be in less than 10% organic solvent. There's a very good reason for that. In reverse phase, if your sample has more than 10% organic solvent, it will not bind to the column. Okay? So if you have organic solvent in your sample, you must evaporate it off first and then resuspend it. You need your sample to be particulate free. That's to protect the column. So you can do that by centrifuging briefly. For reverse phase, you need an ion pairing reagent. In LCMS, MS, you would always use formic acid. You don't use trifluoroacetic acid. That's an ion suppressant. It's a better ion pairing reagent, but it's not compatible with electrospray. And we need a flow rate. Some labs will use nano LC systems. So this is flow rates from 50 to 300 nanoliters a minute. At that flow rate, you need a trap column. Otherwise, it will take you about an hour to get your sample loaded onto the system. Alternatively, you can use micro LC, and that's flow rate of 300 to 3000 nanoliters a minute. And the gradient elution is acetonitrile. Here's a typical gradient. This is for a short elution run. And you can use these. So this is a nano LC column. It's a 75 micron ID column built inside here, and then your electrospray emitter tip is built into the column, and this will plug directly into your mass spectrometry source. So we now come to the data acquisition, and the data acquisition is normally what we call DDA, or data dependent acquisition, or it's sometimes referred to as auto MS to the N. And the problem is you need to tell the instrument needs to know which peptides to fragment. And it's only going to know that as those peptides come in. So you need to program the instrument to make some kind of decision-making process about which peptides are fragmented. So you program that here through this part of the software. So how data-dependent acquisition works is the instrument starts off by doing an MS1 survey scan. So this is our survey scan, and it will identify, as we've shown here, let's say 5 peptides of different mass to charge and different intensities. We then have to have some type of selection, so some type of rule-based precursor ion selection. So you can specify what you want that rule to be. You may decide that that rule may be based on charge state, or it may be based on intensity. In this case, we've chosen intensity, and we've chosen the 3 most intense ions, and these are the ones that we would like to fragment. So first of all, the software will isolate the precursor number 1. Okay, we need to do that because if we're going to do fragmentation, if we have a complex mixture and we fragment everything, we don't know which fragment comes from which parent. So it's important that you can select the fragment, the parent ion first before fragmentation. So that's what we've done. We've isolated the first precursor. We then that is then fragmented, and we generate an MS-MS spectrum. So these are your fragment ions from your precursor 1. We then isolate precursor 2, generate fragments from precursor 2, isolate precursor 3, generate fragments from precursor 3, and then the cycle is repeated again. We go back to the beginning and we do the next survey scan. And this process is known as the duty cycle. That can be 4 seconds, or it can be even less than that. And that is going on all the time. So as you have your LC flow entering into the mass spectrometer, and there are different species coming into the mass spectrometer, all the time this data-dependent acquisition is going through selecting these different peptides for fragmentation. So this is the duty cycle, and this is related to the scan speed. So the shorter the duty cycle and the more precursors you can fragment within a given duty cycle, the faster the scan speed. And as I mentioned in the first talk, the greater the scan speed, the greater the sample complexity that your mass spectrometer can deal with. So let's now move on to data analysis. So you almost always need to do automated data processing in LCM SMS. You will start with raw spectral data. As I mentioned, the data files are very large. You may acquire data over a couple of hours, and you are generating fragment spectra many times a second. The spectra need to be processed and they need to be deconvoluted. And what that means is we will have peptides that may be doubly, trebly charged. They need to be reduced to single neutral mass. Okay, so deconvolution is taking these multiple charge states and reducing it to a single neutral mass. We then need to generate a compound list, and that will contain thousands of peptides in the typical analysis. From that compound list, we need to generate this, which is an Mgf file. Mgf stands for Mascot generic format, and this is a file format which is necessary for database searching. And what that consists of, it's basically a compressed file format, and it basically consists of the parent ion mass, the intensity, the Hplc retention time for that species, and the fragment spectrum, if indeed that peptide was actually fragmented. And then finally, this Mgf file and whatever search parameters you've chosen are exported to the Mascot server. So here's a closer look at an Mgf file. It's just a simplified list of masses and intensities for parent ions and their embedded fragments. So this is the peptide mass, 413.266 to several decimal places. It's singly charged. This is the retention time on the column, 1.5 minutes. This is the intensity of the peak, and then these are the masses and the relative abundances of the fragments. And that's a very simple Mgf file. So when we have an Mgf file, we can now use this to search a database. In order to do that, you have to specify which database you want to search, and you also have to specify rather a lot of parameters, but these might look a little bit complicated, but actually it's relatively simple. So we need to choose a database. A good database to pick if you're unsure is Swissprot. It's a large database. It contains in the order of about 500,000 protein sequences, and this is a good database to search if you're looking for if you're not quite sure what you're looking for. You can specify the taxonomy. A lot of researchers like to do this. They can increase the number of hits they have by narrowing down the taxonomy. Personally, I prefer to keep this taxonomy open. I like to search everything, but that's a personal thing. We need to specify how the protein is modified. So remember, we had to reduce the cysteines and alkylate them, and that modification, carbamidomethyl cysteine, is global, which means that every single cysteine in your sample will be modified. We need to specify the enzyme. In this case, it's trypsin, but you can specify a number of different enzymes, or you can specify your own enzyme. These modifications are variable. Okay, so we have modifications that may be of interest, oxidation of methionine. These modifications, phosphorylation of serine, threonine, or tyrosine. These are variable modifications, which means that if you select that, if your methionine is oxidized, it will be recognized, it will be matched. But also if your methionine is not oxidized, it will also be matched. You need to be very careful with modifications. You can select many modifications, but the more you select, the longer the database search will take. But more worrying than that, the more modifications you select, the greater the chance of a random match. So the greater the chance of a false positive. So for modifications, you want to keep your modifications to a minimum, and you would only select, say, phosphorylation if you had good reason to suspect that your protein is phosphorylated. If you select these for everything, you will find you will get false positives. We're not going to do any quantitation, but you can choose that. We have to specify our mass tolerance. In this case, the mass tolerance is quite large because this is an ion trap instrument. We specified MSMS tolerance. The mass tolerances, so this is the window in which we will find our mass match. We can specify any number of carbon of heavy carbon isotopes. Here we're specifying that we can accept 1 carbon-13, and these are the number of missed cleavages. So we can, in this case, we're selecting up to 4 missed cleavages. You can select more than that. If you're using the public Mascot server, you will find it will probably not allow you to, because the more missed cleavages you allow for, the more search time is needed. We need to specify charge states. This is typical, +2, +3. We specify what the mass is, whether it's monoisotopic or average. I'll talk about isotopes in a later talk, but just to know that when we're searching peptide data, we should always be using monoisotopic mass. And then here we'll specify the instrument type. That determines what types of fragmentation we get. So when we are fragmenting proteins and peptides, the bond cleavage is predictable. Okay, we get predominantly B and Y fragment ions. So this is our parent ion, this is our peptide, and this is the B fragment, and this is the Y fragment. So here's some peptide data, and I've shown you here an example of a very good Mascot peptide match. So what you have here is this top spectrum. This is your fragmentation spectrum. So this is the raw data, and this is the sequence that it has been matched to. So this is an in silico tryptic peptide. We know it's a tryptic peptide because it ends with an arginine, and so this is from the sequence, and this sequence has been generated in silico. So it's an in silico digest done by the computer. This table here, this is a theoretical fragment ion table. So for this peptide, these masses are all the possible fragments that could be generated from this sequence. And some of them are in black, some of them are in red. The ones that are labeled in red, Mascot has matched it to the raw data. Okay, so this B ion series here is present in the spectrum, and this Y ion series here is also present in the spectrum. And this number here, this ion score, this is although called the Mouse score, this is the probability of all of these consecutive matches. So we have a run of 9 fragment ions. So we have a consecutive run of 9 fragment ions, and this is the probability that they can be arrived at by chance. And so that number is relatively big. And what Mascot does is it matches the measured fragment spectra. So the fragment spectrum that you have, so every fragment spectrum that you have is matched to all theoretical fragments of all theoretical tryptic peptides for every single protein in your database. And if your database is Swissprot, that is a very large number of fragments. And so every peptide is matched. So every observed peptide, every observed fragment spectrum is matched to every possible fragmented peptide. And each theoretical peptide is given a score. Okay, so if we have 60 million possible peptides, each one of those is given a score relative to the data that we have. And then those peptides are then ranked based on their Maus score. And the Maus scores are summed for non-redundant peptides from the same protein. So if we have 3, 4, 5 high matching scores to the same protein, they are summed together. And then the proteins are ranked again by Maus score. So this is typical Mascot data, and this is the ranking. So the top hit is this one, FNLA, it's a human protein. However, the protein ranking does not indicate statistical significance. It's a very easy mistake to make. If the protein is at the top of the list, you think, that's a hit. Not necessarily. If you want to know the statistical significance, you don't look at the ranking, you look here at the Mascot score histogram. Okay, so these are the Maus scores for the various things in this table. And in this region, Mascot says these scores are not statistically significant. And in this region, these 3 top scores are significant, but they are the only ones that are significant. So what it's telling you is 1, 2, 3, those 2 hits are probably correct, and the rest of this data is junk. So with LC-MS for analysis of proteins, it's possible to achieve total automation. So you can do that in a setup here. We have, on the auto sampler, we have the ability with 96 roll microtiter plates, we can load 3, so we can load up to 288 protein digest samples. We need to run blanks, so every LC-MS run is preceded by a blank run, so we would do double that number, so 576 LC-MS runs. From that, we would generate 144,000 fragment spectra, and then each spectrum is matched typically against a Swissprot database, which would be 60 million theoretical peptides. And all that is automated. That can be done in 4.5 days with no manual intervention. So we can use that capability in the Centre for Medicines Discovery. We do that. This is our high throughput gel band analysis pipeline. So we start with samples that have been separated by SDS page. We excise those from the gel. We have an electronic sample submission system to speed up that process. We do the reduction alkylation in high throughput. We have a fast tryptic digestion protocol, which takes 4 hours. That's very useful because it means that if samples are submitted in the morning, in the afternoon they can be digested and then it can be loaded on the mass spec overnight. When it's on the mass spec, we do data processing on the fly. So this database searching is done. So the search for sample 1 is done as sample 2 is being acquired. We can then generate a report, and we will do, in the Centre for Medicines Discovery, we will do manual data validation and annotation. This is what it will look like. This goes into our electronic lab notebooks, and the users will then be notified by email that that data is there for them to have a look at. So what we're able to do with this system is we can generate validated MSMS data effectively in real time. I mean, you can get this data in less than 24 hours. At least you could before we had the COVID epidemic. So that's all I want to say about protein characterization by MSMS. Just to summarize, we use HPLC for sample cleanup. It reduces sample complexity. Tandem MS generates protein sequence data. It does not generate, it does not allow you to identify to obtain protein identity. We need to use a specific protease, or we usually use a specific protease, and that's normally trypsin. We've covered the factors that prevent full sequence coverage, and you can see that's actually quite a complicated subject. It's very difficult to achieve full sequence coverage by MSMS. We've looked at instrument programming. We've looked at data-dependent MSMS acquisition. We've looked at data processing. We've had a look at Mascot generic files, and we've looked at basic Mascot search parameters. And then finally, we've had a look at some high throughput analysis. However, this is a whistle-stop tour. We have only scratched the surface, and I would say that mass spectrometry is a practical discipline, and the best way to learn it is to do it. So I'm going to wind up there. Next week, we will be having our only practical session, and this will be an opportunity for those of you who have some sequence data to do your own Mascot search. This will be done online, and it's a very useful thing to know. So that will be next week. The week after that, we have the first of our guest lecturers. It will be Holger Kramer from Imperial College, and he will be talking about quantitative proteomics. Don't miss that. It'll be a very good session. And I am very happy to take your questions now. I'm going to